So we've already heard about the use of early warning systems to help protect against pests this morning, and we've heard about various different risks which are affecting our disease control programs. So I've got 25 minutes to go through how we can use early warning systems to protect ourselves against those risks. So I'm specifically thinking about monitoring and forecasting activities, and I want to give a quick overview of the types of activities that AHDB cereals and oilseeds is involved with. But I also want to recognise where there have been limitations in using those types of activities for a practical purpose, and look at what advances have been made recently and what work is ongoing at the moment to provide you with valuable tools. So as we've heard this morning, disease is affected by three main factors: the host, the variety itself, the disease-causing organism, the pathogen, and the environment. And when all these factors are conducive, we see the development of disease. In cereals and oilseeds, the majority of disease control strategies are targeted towards the host through the use of resistance genes and the pathogen through the use of fungicides. But there are risks associated with all these three factors which affect our ability to control disease. So to pull some of these out: non-durable host resistance, changes in the pathogen population so that they are able to overcome host resistance or become resistant to fungicides, spore release, the spread of pathogens into new areas, and as we heard this morning, climate change and extreme weather events. So in this presentation, I'm going to take a very brief look at each of these individual risks and how monitoring and forecasting can help. So firstly, looking at the risk of non-durable resistance. Now, the obvious place to look for information on varietal resistance is the recommended list. And in my eyes, the activities which are done to create the RL disease ratings are one big monitoring activity. The durability of resistance is likely to be affected by the type of resistance used. For example, if single major genes are used, this type of resistance is likely to not be very durable. In contrast, when multi-gene or polygenic resistance is used based on multiple genes, the resistance is likely to be more durable than single genes, and resistance is more likely to degrade over time rather than break. And we can see these patterns if we look at the recommended list itself. So here, what I've done is to look at the 2012-13 RL and the 2015-16 RL. And between those two RLs, there were 21 common winter wheat varieties. And here we have a graph showing the frequency of each of the Septoria disease ratings for these varieties. Septoria is a disease where we believe disease resistance is based on multiple genes, and you can see that the frequency patterns between the two different RLs are very, very similar, indicating that the resistance has been durable. By contrast, if we look at the same varieties, but this time they're yellow rust disease ratings, where we know that there are major resistance genes acting, we can see different patterns. In particular, if we look at a rating of 9, we see a big decrease in the frequency of varieties with that rating in the 2015-16 recommended list, and also a big increase in varieties with a rating of 6. Now, this indicates that changes in the population have happened which have overcome the resistance genes used in those varieties. But the fact that the varieties haven't fallen off the scale also shows there's likely to be a background of multi-gene resistance in there too. And let's not forget that a disease rating of 6 is not a bad rating. But there are limitations with the information provided through the recommended list disease rating. And the big limitation is that the RL is based upon historical data. The new RL is now available online, and we need to remember that RL contains disease information up until harvest 2015. It does not contain any information on disease in the current season. Therefore, it is really important to monitor all crops for disease, even if they have a high disease rating. So what can we do? Well, within the RL protocol itself, it's very difficult to make changes to bring in-season information. But what we have started doing is additional assessments of disease at some RL trial sites, both winter wheat and winter barley, and we are making information from those assessments available on our website to provide you with in-season information on disease risk and also give you an indication of levels of disease on varieties with particular disease ratings. Sharing information is going to be very important, and social media, particularly Twitter, gives great potential for this. But there are questions surrounding this, particularly, are you guys willing to share your information on disease observations and how that information could be validated? So moving on to the second risk, the risk of changes in disease populations. And here we have an example of phoma stem canker resistance in Australia, where a single dominant resistance gene was used. Now the boxes at the top of the pictures give an indication of the disease population, with the half moons representing spores of individual isolates. The green spores are spores which are avirulent, meaning that they will be detected by the host resistance gene and unable to cause disease. The red spores are those which are virulent, and they will not be detected by the resistance gene and therefore will be able to cause disease. So we can see in 2002, the population was mainly avirulent and there was a healthy crop. But just a year later, we can see the population has changed, so it is now mainly virulent and it has had a devastating effect on the crop. So population changes can happen very, very quickly, especially if pressure is placed upon the population, for example, the use of a single major resistance gene. How can we access information? A good source of information in the UK is the United Kingdom Pathogen Virulence Survey, which has been running since the 1960s and continues to monitor changes in cereal, rust and mildew populations. And there's a range of ways which you can access information from the survey. And I would like to draw your attention to the fact that the team are here today in the foyer, and I would really encourage you all to go and say hello and learn about the survey. And there is also their annual stakeholder event happening on the 8th of March, which will be at the NIAB Innovation Centre, and you'd be able to book onto that through our events area of the website. The UK CPVS provides information on changes in virulence to specific resistance genes, and it also gives information on race structure. In the UK, yellow rust has an asexual life cycle. This basically means it reproduces and produces clones of itself. Every now and again, there is a random mutation event which confers resistance to a resistance gene which has previously been effective. Because of this asexual life cycle, we often see a step change between new and old races with just the addition or switching of a single virulence to a specific resistance gene. And the table here shows that stepwise progression. The exception is the warrior race, which was first identified in the UK in 2011. Initially, it was believed this was the same stepwise progression with an additional virulence added, but since then, molecular genotyping has shown that this race is genetically distinct from those that have come before in the UK, and it is actually a foreign incursion. How do new races affect yield? To answer this question, I've pulled out some treated and untreated yield data from the recommended list for the variety warrior before and after the arrival of the warrior race. So the treated yields are in green and the untreated yields are in orange. In 2009 and 2010, the warrior variety was resistant to UK races of yellow rust, and it had an RL disease rating of 9. There are also insignificant differences between the treated and the untreated yields. In 2011, the warrior race was first detected, but only at a very low level. And in recognition of this, warrior's disease rating fell from a 9 to an 8, but there was still very, very small difference between the treated and the untreated yield. However, just 12 months later, in 2012, when the warrior race had now become established in the population and made up over 60% of the population, we can see the difference between the treated and the untreated yield had increased to more than 2 tonnes per hectare. but these new races can have a massive effect on yield, and as I said earlier, it is really important to monitor all crops for disease. But there are some limitations to doing these types of population surveys. Firstly, the supply of samples, which is a good prompt for me to remind you that if you do see rust or mildew on varieties of a high rating, please do get in touch with the UK CPVS team, and you can find details of how to do that on our website. The costs of doing this type of work are very high, which has an impact on the number of isolates which can be characterised, and it also means that isolates for characterisation tend to be chosen from lesions which appear on believed to be resistant varieties, when in actual fact, changes in the population are perhaps more likely to be recurring on more susceptible varieties. There are also issues over the timeliness of information, as the results can take a long time to come from these types of experiments. But advances are being made, and molecular techniques are going to be key to this. There is a new concept of field pathogenomics, which has been developed by Diane Saunders at the John Innes Centre, whereby rather than taking samples through traditional pathotyping techniques, they can be sequenced to give information on genetic diversity, and also you'll be able to identify the variety from which a sample has come. We have a project running at the moment which is aiming to develop a pathogenomics platform which can be inserted into the UK CPVS survey in the future. But in the meantime, we have made some improvements, and the UK CPVS team have been able to change their reporting structure to get results out to you quicker. As I mentioned earlier, we've been doing additional monitoring of RL trials to ensure that we pick up any changes on our own trials at an earlier opportunity. And again, social media has massive potential for sharing observations of unusual levels of disease on specific varieties. So moving on to the next risk, and this risk of spore release. There is a direct link between spores and disease, and the graph here shows how the incidence of Sclerotinia disease increases the closer you get to a source of spores. Air trapping and monitoring of spore release has been used as a research tool for a great many years, but there have been problems with using this practically for giving alerts of disease risk. And this has largely been because of the methodology, which has required examination of air samples under the microscope. This has needed a trained fungal taxonomist. It's taken a lot of time, and it has not always been possible to distinguish between closely related species. Recent advances in molecular biology have allowed a huge step forward. For example, it is now possible to distinguish between closely related species. Here we have a graph showing the spore release patterns of the two phoma species, Leptosphaeria maculans and Leptosphaeria biglobosa. And you can see Leptosphaeria maculans, which is the orange line, has an earlier ascospore release than Leptosphaeria biglobosa, and this helps to explain why lesions and cankers caused by maculans are more severe than those caused by biglobosa, as the spores can infect younger plants with smaller leaves. It's also possible to look at traits using air samples. And again, here the example is phoma, and this is where molecular techniques have been done on air samples to quantify levels of avirulence to specific phoma resistance genes. The orange bars there show the results of traditional pathotyping work, which was done on isolates collected from the same field. But we do need to remember here that with spore trapping, you're only able to identify what you're looking for. You can't identify the unknowns. And you also have to remember when looking for traits that you're dealing with a mixed population, so it's very difficult to make conclusions about race structures. Now, if we look at this graph, we can see Sclerotinia spore release patterns in the orange from a field site and in the green from a rooftop site, actually in Worcester Town Centre. And you can see that the amounts of DNA are very, very different, but actually the pattern is very, very similar. And this shows the huge potential that spore trapping has to give in-season alerts for disease. And indeed, it is being used in programs such as the Brassica Alert Spore Trapping Network. So to recap on limitations that remain, timeliness of information is really important here. The picture shows a 7-day air sampler, which has traditionally been used for spore trapping experiments. And as you can tell by its name, 7-day air sampler, it takes in excess of a week to give results. But advances again are being made. This is an automated spore trap which has been developed by Rothamsted and Burkhardt, and this spore trap can detect and quantify spores rapidly in the field. We have research going on at the moment to refine this technology to be used to detect Sclerotinia, Septoria and Fusarium spores. And the spore trap is already being used as part of other alerts, for example, the Blight Alert. So moving on, and we've already touched on this morning, the risk of pathogen population spread. Those spores can travel huge distances when picked up in air currents, but there's also risk of population spread from the transport and movement of people and goods. And this map shows the major transportation routes around the world. And I'm not able to point out the UK, as you can't see us because we're under a glow of air, shipping and rail networks. Some work's been done recently to look at the global spread of yellow rust. Now this really deserves a presentation in its own right, and I will try to quickly explain the results. So a worldwide collection of isolates was examined genetically, and the genetic results were used to split isolates into different groupings, and then hypotheses made about how these groups of isolates were able to travel throughout the world, with both air currents and the movement of people and goods being indicated as the main transportation devices. But what you can also see from this work is that in the area around the Himalayas, there was a lot of genetic diversity. Now, as I said earlier, in the UK, yellow rust can only reproduce asexually and produces clones of itself. In the area around the Himalayas, yellow rust is able to reproduce sexually, leading to a high level of genetic diversity, and this may well be the source of new races. So when we're thinking about risk, we really shouldn't be too short-sighted at just looking at what's happening in our own country. We need to take a global perspective, and we need to work more internationally on collaborations. For example, UK CPVS contributes results to the Global Reference Centre. International collaborations could also help us in terms of learning how other countries deal with disease and help us to refine our own research methodologies. So moving on to the last risk now, climate change and extreme weather events, which I won't go into really because we heard a lot about that this morning. Flash you some news headlines, but you've all seen the news today, so I didn't bother with that slide. But we can use the weather to help us forecast and predict disease. And here we've got an example of how a weather-based model called ScleroPro has been used to forecast Sclerotinia infection events. And a few years ago now, we have a project with ADAS which looked at comparing fungicide control achieved through applying products according to the ScleroPro infection alerts in comparison to standard fungicide timings. And you can see from the chart that the ScleroPro infection alerts gave as good disease control as the best fungicide timing, showing the huge potential for using this model practically. But we have to think back to the disease triangle. When we're using weather-based forecasts, we are only using environmental information to predict disease. Disease isn't just dependent on environmental factors, it's also dependent on the host and the disease itself. So can we improve weather-based models by adding in these extra factors? And for Sclerotinia, this has been done. So the environmental factors are still there in the form of the ScleroPro model, which we know works really well. Additional information on growth stage has been added, as well as information on the pathogen in terms of information on the presence of spores and of infected petals to give an overall disease risk, which is now available on our website during oilseed rape flowering. We also have a network of weather stations which are operating at RL trial sites across the country. The data from these stations is available on our website, but it's also being used by disease forecast models. Now, over time, the data that these stations are creating, along with the observations done through the RL at the same sites, is creating a very, very valuable data resource. So in the future, we will be able to make better predictions about the effects of extreme weather events by looking at this data set. So in summary, in terms of monitoring and forecasting for disease, we have a lot of balls in the air. What we need to do is look at ways of sharing this data, both within the UK agricultural industry itself, also internationally. There is also a need to start integrating this information rather than using them all separately. And as a first step to this, we have moved all of our disease forecasting and monitoring to a single web page, so you only have one web page to go to get all of this information. And you can sign up for an alert service as well, which will alert you if anything new goes on that page. So to conclude, we're facing an uncertain time for disease control. Forecasts and monitoring have been around for a long time, but there have been factors which have affected their use for practical purposes. Recent advances, particularly around technology, monitoring, data sharing and integration, have allowed us to improve our services which were currently available. We've brought all of our services now to the one web page, so you have one place to go. And you can sign up to pest and disease alerts. And my final thing to say is really, this is such a fastly evolving area. Really watch this space, and there could be several more improvements coming in the near future. Thank you.